So hello everyone again. Welcome again to another Saturday live stream. Microbehunter here. Today's topic I would like to show you again my stereo microscope, and I'd like to put some specimens under the stereo microscope. And yeah, we're going to see where this is going to take us today. Well, I would like to, as always, kindly request to give me a short thumbs up if the sound is okay or not. Because sometimes there is a little bit of distortion, sometimes the sound is not loud enough, and yeah, but the first message is here that the sound is okay. Yeah, excuse me, I see that some people already started to write comments in the comment section, and yeah, if you have any questions, of course, then I'd like to answer them. Well, I have to admit to you that preparing for this live stream was a little bit technically challenging because I had several USB issues. Having so many cameras connected, I had some problems getting a stable connection, and I hope that, yeah, for example, here's now the table view. Yeah, I hope that everything's going to work out fine. And for those of you who joined in last week, you probably remember that after a couple of minutes, for whatever reason, the live stream was interrupted and I had to start a new live stream. So I hope that this is not going to happen, but if it does happen, I just want to inform you that it will then start a new live stream, but it will be then yeah, a completely separate video. Okay, so it will not be a continuation of the live stream that's currently in running, but I have to then start a new one. Okay. Yes, very good. Many people already are saying hello. Okay, if you have any questions specifically for me, then please write it in the chat with an @Microbehunter or @Oliver, so that I know that I'm able, that I'll basically respond to it. Again, hello from all over the world here. That's very nice. And what I would like to do first is, yeah, I would like to first show you a little bit my stereo microscope that I have, which I received yeah, a couple of several weeks ago. Yeah, you can already see also in the above me that this is basically also a view here. And I've attached a ring light, and let me just move it away a little bit further. Yeah. This is how it looks like, and you're probably already wondering why I covered the eyepieces with a little cloth. And the reason is because if I do not have a cloth over it, then the light from my room will go in here, and it will actually reflect, and it will actually reduce the image quality. Yeah. So it's the light from the lamp, yeah, going in here, and then yeah, it decreases the contrast. Yeah. So that's the reason why I covered it up here. Here, up here is my USB camera, which I think is a little bit slow. Okay, it's only USB 2.0, yeah, and it's directly connected, and that's basically what you're able to see here now. Here, I also adjusted everything so that it's auto exposure. So this basically means that it might take yeah, a couple of seconds or fraction of a second for the camera to adjust its exposure time a little bit. Okay. So that's basically what I would like, you know, where I'm going to show you a couple of things here. And before I do that, I would like to also show you just very quickly, I always have to switch over again to the desk view. Yeah, so-called Barlow lenses. So some of you might already know about those because I showed them already in the past. But about a third one. So this is, these Barlow lenses can be attached beneath the stereo microscope, and this increases the magnification by 2 times. This reduces the magnification, and the purpose is that you then get a greater depth of field and a larger distance between the specimen and the objective. Yeah, so basically you can move it further away. And the third one that I've got now, and this is the one that I've currently connected, is a 0.7 magnifying Barlow. Here, let's go here. And this allows me to have a greater distance now between the specimen and the and the objective, and also it makes the image a little bit smaller because I do not need large magnification. The camera itself also magnifies a lot, so I actually do get a slightly better image quality this way as well. Okay. So, and then beneath this 0.7 Barlow, I have another ring which is able to hold the ring light here. Okay, so this is a lamp that I've added later on. Of course, the microscope also has a built-in lamp over here from the top. It also has a lamp from the bottom here. Okay, but I'm mostly using the ring light because it's quite powerful and it gives me enough light. Okay, so this is kind of the setup that I'll be using today. Yeah. By the way, there's also knobs here in the back. That's for controlling, of course, the light from the top and from the bottom. Okay, if you're interested in this microscope, I did make in my other channel in Microbehunter Microscopy, I did make a review of this. It is from the company Euromex. It is a zoom stereo microscope, so by turning this here, you can see that I'm able to zoom in and zoom out. And of course, the more you zoom in, the darker the image becomes, but the camera, as you can see, it's only dark for a very short time, and then have to refocus. The camera essentially always readjusts the brightness. Okay, so just so that you a little bit understand the setup that I'm using here. Okay, stereo microscopes are often a little bit neglected. Most people who are into hobby microscopy have compound microscopes, and compound microscopes allow you to look, of course, at much smaller objects as well, but that's actually one of the reasons why I said, okay, let's do a little bit of stereo microscopy today as well, so that this is also not completely forgotten. Okay, so as always, every couple of minutes I'm going to interrupt myself because I would like to read a little bit of what's going on in the chat. Okay. And there is already a question here. Can anyone tell me what the stereo microscope and how it's different from a normal microscope? Okay, that is actually an important question. When you're into hobby microscopy, you have a choice of different types of microscopes, and stereo microscopes, they basically have two eyepieces. I'm just going to go up here a little bit. Okay, so two eyepieces, one for the left and the right eye. But the two images that you get are different. Okay, because there are two separate light paths down here, and there's one objective from the left and one on the right side. And I don't know if I'm able to show this to you. I'm just going to try this. Are you able to see this? I don't know if you're able to see this. Oh, no, it doesn't work. Yeah, on the top, just right here on the top, you might be able to see two objectives. I have to focus this, or maybe I have to turn down the light a little bit. Yeah, here. Yeah, are you able to see those two little on top? Okay, you might see those two circles in there. Yeah, these are the two objectives. One for the right eye, and two lenses. Okay, one for the right and one for the left eye. Okay, and this basically means that the specimen you get two different images. Okay. By the way, I'm just going to show this. Do you actually see this? Look on the side over there. There's this bright spot. Okay. Look what happens when I cover the eyepieces now with this cloth. Okay. Yeah. It looks much better. The reason is because it's a stray light. Okay. It's a stray light coming in through the eyepieces. That's why I'm covering this. Okay. With the cloth here. Okay, so then you get a better image quality. Now I have to turn up the light again here. Yeah, so that is basically stereo microscopes allow you to look at objects that are opaque. So basically, like this leaf over here. Okay, maybe I don't know. I'm gonna show you later on. Some lichens here on the wood. Okay, and some solid objects, and which are not transparent, and they magnify quite large. Not as much as compound microscopes, but you get a stereoscopic view. This means actually you get a sense of depth. Yeah? With compound microscopes, you have to use microscope slides, like I've got these here. Okay, and the object that you can look at has to be much smaller. Yeah? So these are basically two essentially different types of microscopes, and essentially, it depends really what you want to look at. Yeah? Generally, if you're really deep into hobby microscopy, then you're probably going to have both of them sooner or later, but it depends really what your interests are. Okay, so but because stereo microscopes are not quite as common in the hobby microscopy sphere, that's the reason why I decided to do another one with stereo microscopy. I like to know more about the use of stereo microscopy in histology. Every time a biopsy is obtained at my hospital, a biomedical scientist is checking the specimen under a stereo microscope first. Okay, so it could be that what you do sometimes is you can actually take a slide like this and then put it under the stereo microscope for a quick quality check. However, do not confuse stereo microscopes with binocular compound microscopes. Okay, so compound microscopes that have one objective, they basically also can have two eyes, like mine, for example. I'm going to show again I'm going to turn around the camera here, right? Okay. Yeah, so that's my other microscope that I have, yeah, the compound one, but it also has two eyepieces, yeah, but it's not a stereo microscope, and therefore some people like to confuse those two, okay. I know stereo microscopes are very often used to do quality checks. Are air bubbles present, for example? Okay, is the specimen flat, and so on. Yeah? So this is basically the reason. Yeah? So basic as a compound a comment again. So basically, stereo microscopes allow us to see 3D. That's correct. Yeah, you can directly see objects. That's correct. So and for normal microscopes, it says here in parentheses in the comments, which are light microscopes. Well, stereo microscopes per definition are also light microscopes. Both of them are light microscopes. Yeah, and so that's the thing. Yeah. The two objectives are not good to see because of the Barlow lens. This could be the reason is because there's an extra optical system in there. I mean, if you want to, okay, why not? I'm going to do now the following. I'm going to just show you. I'm going to disconnect this. Okay, I'm going to disconnect this and I'm going to show it to you again. So that's why it's dark now. Okay, just disconnect this. You have to now look at what I'm doing here on the top. It's a little bit small, I just realized. You know what I'm gonna do, just for the fun of it? Let me try something. I would like to do the following, just a second. Look, is this gonna be possible? I'm gonna try to make it bigger. Let's make it bigger. How is that? I think, okay. This is a little bit better now. So this here, there is a ring down here, which is for connecting the ring light. Okay. Let's move over here like this. And this is the Barlow lens, yeah, which I'm going to connect the ring again and okay. And I'm going to show you again yes, you see that's the, yeah, makes everything a little bit smaller. So I've got 3 of those now, yeah. And the reason why I'm not using the 0.5 one is because then I'd have to move up the microscope so much that I'm not able to focus anymore. Okay, so this is a little bit too much already. So I'm going to show you now, or I'm going to try to show you now the two objectives. So I'm going to flip the camera around. Yes, here I think it's much better. Okay. Okay, I have to get some, yeah. So you see the, let's turn on the light. Yeah. Yeah. Okay. Yeah. Let's see what happens when I zoom. So I'm zooming now, so you can see maybe the optical elements move in and out. Okay. Yeah. So that's basically the thing. Yeah. And then here, of course, you can see the two eyepieces. Yeah. So there are basically different stereo microscope designs. This is the most common design. There are also stereo microscope designs where it's called the common main objective, the CMO design, where there is only one main lens on the bottom, and then both eyepieces basically use the same main lens. I think those microscopes might be a little bit more expensive because you need larger optical elements. Yeah, and let's go back again here like I had before, and I'm going to move this again like here, and I'm going to now reconnect again okay, I have to, just a second, I have to attach first this here again, and then let's continue observation. Okay, but I just wanted to show you a little bit, yeah, the microscope here as well, so that you essentially know, yeah, what you're looking at. So, and then I'm going to connect this here again. Everything takes a little bit of patience. During a live stream, you have to also improvise as well a little bit. So here we go again. I hope it's tight. Okay, let's turn it on. No, yes, and the light. Here we go. By the way, this is moss. Okay, so that's a little bit the thing, so that you know, yeah, what we're doing here. Okay, so. Much better to see the objectives now. Thanks for the extra effort. Yes, yeah, I think we're all here to learn something. Okay, so that is the moss which I collected today, and yeah, my hopes were not fulfilled. I was actually hoping to find some tardigrades, but they are indeed small, and yeah, maybe yeah, the sample is not the very best one, but of course we are able to look at yeah, at the moss here. So what we see, however, is the following. Also something that I would like to clarify a little bit here. And let me turn on the arrow. So I have a nice little, a tiny little arrow here that I'm able to move around, okay? And you might see those circles, okay? Those round circles, these are, because there's water there, these are reflections of the ring light. Okay, so these are artifacts. So that's something that you have to take into consideration a little bit when using those ring lights. And what I'm going to do now is I want to show you how the reflections look like when I switch off the ring light, okay? And let's switch on the light from the side, okay? So that is now this lamp over here, okay? Yeah, you can see that it's now the light coming from the side. And this, of course, changes the entire characteristics again, okay? And you see that the reflections also look different right now because the light is now from a point. So that's certain things that you might want to consider when observing things under the microscope, because sometimes, I mean, you do need light, but sometimes the light that you're using or the lamp that you're using actually causes reflections which you might not like. Okay, so I'm just saying. So let's have a look now how the whole thing looks with light coming from the bottom. Okay. Yeah, and of course, again, different qualities, and because the moss is relatively dark, yeah, therefore you can see the camera adjusts its exposure time. Yeah? Therefore, it looks again different. Yeah. Of course, if you want to, you can use also a combination of things. So, for example, you can of course also use light from the top and from the bottom. So it depends a little bit what you want to do. Okay. So it's now from top and bottom, and then when I switch off at the bottom, it's over here. Yeah, you see that it again looks different. Okay, just saying. Yeah, there's somebody commented, I sent you an email for advice on buying a microscope. Yes, I receive a lot of emails. Sorry if I'm not always replying very immediately. Yeah, I'm kind of trying to work off my email list. Yeah. So very important also is the following when buying a microscope. You see, many people ask me which microscope should I buy, which brand should I buy, and so on. And I have to tell you, that is one of the most difficult questions because the expectations are so different. Okay. Generally what I say is don't spend too much at the beginning and kind of upgrade as you go along. And with upgrade, I mean not necessarily upgrade the microscope, because many microscopes have limited upgrade possibilities, but rather buy yourself a slightly better microscope. Okay. So one of my favorite specimens I'm going to show you now and as well, and this is a fly which I caught some time ago. I always zoom out. Okay. This is a fly, which and of course you also have to, you probably have already seen it before. This is a pretty large fly, and I'm going to go up with the brightness again here. See, and the camera will readjust. Okay, so I basically put it on a little piece of cork. Yeah. And we can now zoom in a little bit, and one of the things that's always interesting is to look a little bit at the depth of field. I'm not going to do any focus stacking right now, but it is possible to combine the focus together so that you get an image which is in focus all the way through, right? But as we zoom in, okay, and it's not properly adjusted in my microscope, because normally even when you zoom in there should not be a necessity to actually refocus. But what I like, wrong direction. What I like to see here is look at the eyes. These are the so-called the omatidia. These are the so-called the eyes of an insect. So it's these are the individual elements. So an insect has compound eyes, and with a stereo microscope we are able to see this actually quite nicely. And why am I showing this to you? Because this actually illustrates that there are certain things that we can see very easily with a compound microscope, and which we're simply not able to see with a stereo. All the way around. other way around. Just simply things that we can easily see with a stereo microscope, which we cannot see with a compound microscope. Because if I want to put this under this slide, under the microscope as it is, I'm not going to be able to see anything. It's just going to be black because, of course, it's not thin enough and I cannot fit it on a microscope slide. So I'd have to make cuts and sections and so on. This is actually the reason why I say that compound microscopes and stereo microscopes, they complement each other. And each microscope has its own world, so to say, that you can observe and look at. Okay, so I think okay. So this is a little bit the things that I wanted to show you, and of course, if you we always zoom out again. And by the way, as I turn, it's also something I want to show you a little bit here. I can, as I turn, it clicks in. Maybe I don't know if you're able to hear this. But it clicks. And this is actually used for making measurements, and for doing calculations. So this is the thing here. So let's here, for example, this is the cork. Also a very suitable specimen because it's very flat. Okay, so. And of course, we are not able to see the individual cork cells because they are connected together so densely that this might be difficult to see. Okay, and focus. So I also want to show you something else right now. Look, as I'm focusing and as I'm moving the microscope head up and down, look, this string that you see over there on the side, this long thin thing, that's some kind of a dust fiber. But look at the position of this. Just let's use this as a reference as I'm focusing up and down. Of course it goes blurry, but what else do you see? Do you actually also see it moves left and right? That is completely normal. Okay, and the reason is because the camera uses only one of the objectives. Now I don't know if it's left or the right one. So I'm covering now the right one. Now I'm covering the left, I don't know, it only uses one of those objectives, and therefore because those objectives are not aligned at the center but left and right of the axis, therefore as you move up and down, it kind of the image appears to shift left and right. So if you want to do some focus stacking, for example, in the software, it will actually also align the different images together. But I just want to say that this is completely normal, that if you focus up and down, that the image moves left and right in the camera. You're not going to see this, of course, when you look through the microscope with both eyes, but it's only an effect that happens over the camera. A great view of the fly's eye. Can you estimate the enlargement? As a matter of fact, almost. The thing is the following. I'm going to just show you what I have here is the following. If you look over here at the magnification, okay, if you look over here at the magnification, I don't know if you're able to see this here. It says here 5.5 times, and then what you have to do is and it goes all the way to 1 time. Okay. So, and the magnification as you look through the eyepiece is calculated as follows. 1, then the eyepiece magnifies 10 times. Okay, it says here 10 times. So it's 10 times magnification, but you have to multiply it also by the Barlow lens, 0.7. So the magnification when looking through the eyepieces is now 7 times. But I can go even further down. Okay. The lowest is here, it says 0.65. I don't know if you're able to see this, it's a little bit out of focus. I have to focus it. So 0.65 times 10 for the eyepiece would be 6.5 times without the Barlow, but then you have to multiply it with the Barlow. Okay, so that is essentially the magnification as you look through the eyepiece. So this microscope, actually, if I zoom in all the way with 55 times, okay, 5.5 times 10 is 55, and if I were to add now the 2 times Barlow over here, if I were to add now the 2 times Barlow, then I would have 55 times 2, well, over 100 times magnified. If you want, I can actually also try that, but I tell you, it's the depth of field is so low that essentially the quality is also very low. Yeah. So, but I essentially just wanted to quickly answer this. Yeah. So another question: if microscopes and telescopes merely photoshopping images to compensate for what the naked eye can see, do they actually see far out the inner space, or do they actually see far out or the inner space? If I understand correctly, well, let's put it this way. I hope I understand your question correctly. If basically microscopes and telescopes, of course, can only magnify that what they receive. Okay, so the information that's not there, they're not able to magnify. Telescopes, however, are not really seen as magnification devices. Telescopes should be seen as light gathering devices because they are actually collecting light which is too weak for our eyes to see. Okay, and of course they also magnify, but actually what is really important is that they collect light, and that's why the diameter is very important. And for microscopes, essentially what we have is that they allow you to see objects which are too small because our eye is not able to focus close enough. So in other words, if I want to look at here, I've got a leaf and I want to look at the cells of this leaf, then I'd have to basically move it really close to the eye, but there is a certain limit where the eye is not able to focus anymore. Okay, and for this reason I'm not able to see it, and for this reason you need magnification devices like microscopes, so that you're essentially able to move much closer than what your naked eye is able to perceive. Okay, so I don't know if this kind of answered the question. So so this basically is the thing here, and this is basically one of those nice specimens that I've got. Which has been now here for, I don't know, 2 months maybe. Yeah, look here. But it's a gigantic fly. I've never seen such a huge fly before. Okay. Yeah. So so if the light isn't there, it doesn't fill in the blanks. I don't know if I understand you correctly. I mean, if the light isn't there in the first place, well, I mean, there has to be a little bit of light there, and then the microscope or the telescope or whatever is able to how do you call this? Make it stronger, bundle the light, focus the light, so that we're able to receive it. But if something is not there in the first place, then no technical device is able to create that information. Okay, so this is a little bit the thing. So what's not there is cannot be caught. So for example, I think this is also quite important here when looking at stereo microscopes and so on. If I want, I mean, I can zoom in, okay, but there is only a certain limit, and from a certain point onwards, it's going to become blurry, because essentially the optics is not able to gather the information anymore, and then it becomes blurry. So let's move on, because I do still have a couple of specimens I want to show you. This here, how many of you know what this is? If I have to kind of zoom out. It was actually still alive, but it stopped moving a little bit. This is a silverfish, it's called. And it's an arthropod. So you have to go all the way down again. Maybe go up a little bit with the light intensity as well. They can be found in some homes, and I was lucky enough to find one. Not everyone likes to see those. It stopped moving. Here. It's still alive. Okay. They're completely harmless. They eat all sorts of things. It's not able to move up the petri dish here. Maybe I woke it up. It would have been better maybe if we would have kept quiet. Let's have a look. Let's go in a little bit. Okay. And yeah, that's of course now difficult because it's not, yeah, it's trying to crawl upwards. And we can see again that the camera tries to compensate the exposure time. Let's zoom in even further, and we're able to see the scales. That's one of the things that, yeah, and a tiny little hair. Yeah. But you see again here at this magnification and so on, yeah, it's already very dark, and the camera compensates the exposure time, and then it becomes a little bit all washed out. Yeah. So. And here we go again. Yep. These are silverfish. Arthropods, of course, feed on, yeah, I guess, skin cells that can be found in the dust and so on. Okay, completely harmless. And are not known to transmit any diseases, but some folks don't like them. Yeah? So you have a variety of of species around. Yeah. Yep. Okay. Yeah. So that's basically something here as well. I want to show you something else now. If you look at the background here, let me zoom out again, or maybe you want to just look at it. Yeah, on top. I need to turn this down a little bit. You can take this plate out, okay, and there is an LED lamp down here which you're able to see now. Okay, so there's this plate, and you remove it, and this is LED there. Okay, you can also, of course, put a plastic plate in here for, show it here. It has a white and a black side, and the black one on top, so that you hopefully are able to get a better or a different contrast. Okay, but I found out that actually, yeah, even the plastic, even the black plastic reflects quite a bit of light. So is there not a way that we are able to somehow make the background completely dark? And I did find a way, a little bit by accident. What you do is you place a mirror on the bottom. Okay, so that's a mirror. Okay. And you place the other one on top, and now you actually see a little bit the dust much better. Yeah. But this also gives you a darker background. So let's try the following. Maybe we can just turn on the light from the side a little bit, not too much. Yeah. And you might also notice a little bit that the camera itself is not, you see it's a little bit laggy and not quite as smooth. This has to do something with the exposure time, and also the fact that the camera is a USB 2 camera and therefore a little bit slow. So I just wanted to show you here that there are a variety of little tricks and things that you can try out to get better images. And that all of the dots that you see here, that's all dust. So there's the petri dish itself was not quite clean. Yeah. Is there a relationship between working distance and depth of focus? The more working distance, the less depth of focus. Well, actually, the larger the working distance is the larger the depth of field. Okay, this has to do also something with the diameter of the front lens. You know, the thing is ideally, but this is of course not possible. If you want to have almost infinite depth of field, you have to have a lens which is infinitely small in diameter. Then of course you don't get any light through it, right? So it's a little bit like this. Imagine if you're looking at at objects which are very far away. Okay, I don't know. Let's say a tree and a mountain, several trees and mountains and so on, very far away. Then essentially everything is going to be in focus. Okay, maybe the mountain which is standing in the tree which is standing in front of the mountain, maybe they're quite far apart, but both of them appear to be in focus, right? But if as soon as objects become near to you, the depth of field becomes more narrow, right? So and this is the reason why I actually added this Barlow lens here, is because I'm moving further away from the object. Okay, and therefore I also get a larger depth of field, and which makes it easier for doing YouTube videos and demonstration work and so on. Huh? So that's the That's a little bit here we go again. Okay. Yeah. Yeah, so just a little bit, yeah. So another thing I just wanted to show you, also something which I've been trying and experimenting around with, but not fully successfully. I just want to show this to you, and that is, I'm going to just, before I show the other things, I'm going to go again to the desk view. Where's the desk? Here is the desk view. Yeah, see, I've been talking so much that I didn't even have time to show you some of the other things here. So this is essentially a, yeah, many of you probably already know that it's the mobile phone adapter. I bought myself a separate eyepiece here, okay? And in here you can put your mobile phone, obviously, right? And when you do that, yeah, then you have to line up everything properly, and then you can either put it into the eyepieces, but not in a stereo microscope, because the stereo microscope eyepieces have a larger diameter. But I can put it in into the folder tube on top. However, those systems have some huge advantages and huge disadvantages. The huge advantage might be a little bit surprising to you, and that is that the image quality is pretty good. As a matter of fact, I think the image quality is better than the microscope camera that I'm using, because I'm getting a very large field of view. So basically I see the whole image in a circle, while this camera adds an additional magnification to it, and this causes everything to be a little bit more blurry. Yeah? So I think those mobile phone adapters and so on, they are pretty decent, and I would actually recommend that you try those first. However, they also have another huge disadvantage, and that is stability. Right? If it's a little bit wobbly, okay, if there's only a tiny amount then the image is already of a much worse quality. Yeah? And what I've done therefore is I've 3D printed myself this plastic ring here, which I can put in here. Oh, I'm just going to show you here. Yeah, I can basically put it in here over the eyepiece and the camera to block any light. Yeah. That might go in, yeah? So that's kind of the thing, and it also gives it a little bit more stability here. But alignment is indeed a problem. So and what I've actually also experimented with, but then I have given up on that, yeah, it is possible to connect this camera over USB or even over wireless to a computer and to use it like a webcam. So theoretically, it's possible to use a system like this also for doing, for example, live streams. Yeah. I tried setting that up, but found out that the disadvantages of the system being very unstable, yeah, I kind of decided, okay, I'll accept a slightly lower image quality with the microscope camera that I have here, and I'm simply going to live with that, so to say. Because the stability is much higher in the microscope camera. Yeah? But again, because it does add a little bit of magnification, therefore the image does become a little bit more blurry. Okay. Yeah. So can we focus on its face? Okay. So that's essentially the question about the little how do you say? Yeah, it does have a nice face indeed. It also has compound eyes. So I hope it's now a little bit at rest. Yeah. Oh my God, he's got an Android phone. Well, it's an old Android phone. Yeah. And as a matter of fact, the Android phone does have the advantage that I'm actually able to directly connect it over USB to my computer. Okay, so this is a possibility. I can convert. There's an app that you can install, or the several apps that you can install, and those apps allow you to use Android phones like a webcam. Okay, and therefore it is actually possible to yeah, also use it for streaming. So that's basically, yeah, that's basically, so to say, the face of this little silverfish. Okay, I'm now reaching the maximum magnification, and I have to maybe, yeah, I'm also going to turn up the light on yeah, from the top. See, there's a lot of stray light as well, and that's already the maximum that it can go. Yeah? So a little bit like this. Yeah. Yeah, you can see the individual scales, everything. Yeah. Yeah. Okay. Yeah, of the tiny compound eyes and everything. Yeah, it looks kind of Now it's moving. Yeah, and the camera is of course always readjusting. Yeah. So that's this one here. Those stereo microscopes. We also, you know that I'm a teacher, of course, in school as well, and we are using those stereo microscopes actually quite a lot, especially with the younger students, because there is very little preparation time needed. All you do is you take an object, whatever you want to look at, an insect or whatever, and directly put it under the microscope, and so it's a very straightforward and direct way to connect students with their immediate environment. Yeah? So, polarized light and analyzer works great with stereo microscopes. I'm just reading, yes, look what I've got here. Polarization filter. So, I've actually, I'm not so happy with the vitamin C slide here. Okay. Yeah, but I'll put this under the microscope as well. Can you please tell us the make and model of the stereoscope you're using? Yes, I can do that. The company is called Euromex, and this one is the Nexius Zoom Evo. And I did make a review video of this microscope. Okay, and that's the Nexius Zoom Evo. There is a whole, I mean, they have a whole range of Nexius Zooms with different stands, and it's configurable. Okay, so I use this stand here, but there are different ones as well. So if you actually go to the homepage of Euromex, you'll see everything there. And if you want to have a more detailed review, in my other YouTube channel, Microbehunter Microscopy, if you go there, you can actually watch the more detailed review of this stereo zoom microscope. Yeah? However, there are of course other brands as well, which are quite nice. Yeah, but I like this one quite a lot. I have to tell you, it's actually I'm very satisfied with this. Yeah, yeah. See, it's a little bit too dark already. Yeah. Silverfish are interesting in that they digest cellulose via bacteria and that they do not require external water. Yes, basically, let's do a little bit of biology. For example, humans we are directly not able to digest cellulose, even though cellulose is made of glucose. That would be a very interesting energy source to use. However, we are not able to digest it because we don't have the enzymes. However, there are bacteria also, for example, in cows, which eat a lot of grass and cellulose, and certain arthropods here as well, where there are bacteria that do have the enzyme and they're able to digest it. And how can they actually survive without water? Well, by cell respiration, by oxidizing organic substances. And essentially, they are able to make their own water. And as a matter of fact, this is also the method that some other animals use who are able to live in the desert. Yeah, by burning up organic material, by oxidizing it, but in the process of cell respiration, water is created as an end product besides carbon dioxide. Yeah? So this is one of the ways that these critters are able to do that. You know what? Let's move on a little bit. Okay. What time is it? Oh, 43 minutes. Yeah. The other thing I want to show you, I did not look yet. Was having a walk today and I found this branch here with some lichens on it. So let's have a look at this. It's very dry. Occasionally you also find some water bears, but not in this condition here because it's in a dry state. Yeah. So so these are lichens on a piece of wood. So what I could actually do, and I've done this before already, if you soak it in water a little bit, if you wait a couple of days, then whatever is on here comes back to life. Also a whole bunch of nematode worms and amoeba and I don't know what. Yeah. So it's going to start living quite a bit. Yeah. So and see the lichens, and let's completely zoom out first. So that's basically you're not able to see the top here. Okay, so let's do it like this. So maybe you're able to see this not better. Okay, here we go. So that you get a better orientation of what's going on. And let's start zooming in. How far can we go? Yeah, you hear it click maybe, so that's the click stop. And it's already pretty large. Okay, and then you focus a little bit. Yeah. So this is but I think I always think that this magnification is a little too high. Yeah, of course, if you start exploring the environment and nature like that, then you might of course find tiny little mites, insects, whatever, as well. Yeah. By the way, last week I found several lacrimaria in my moss sample. Oh, that's interesting. I remember that lacrimaria are your favorite. Yes, I have to tell you, lacrimaria, they are protozoans that have this long neck. Yeah, and I have to tell you, I did not. Maybe I'm not looking always at the right places, but I have not found them so often. Yeah, and that's maybe one of the reasons why I like them. I found them a few times, but not quite as often. Yeah? You know what? Just in case you're interested, but I need to maybe change a little bit the specimen here, because this one is not flat enough. I want to show you a little bit the how it looks like if you actually add the 2 times Barlow. Okay, so this basically means we're going to go above 100 times magnification, and don't have high expectations. I just want to demonstrate this to you, that maybe the image is not even useful at that magnification anymore. Okay, but I just want to show it to you. Yeah, so I'm going to take this off here, the lamp. Okay, so here I'm going to take the lamp off. I'm going to switch it off maybe as well. I'm going to move this back a little bit so that you see again what I'm doing. Yeah. It would be nice to be able to record from both eyepieces and then look at this with VR Goo. Yes, this is actually also something I've been thinking of. You need kind of 2 cameras maybe from each eyepiece, and then kind of combine the image together again. Yeah. So I'm going to remove the 0.7. I have to also remove I'm going to switch over to the desk view again. Yes. So that is, this is the adapter ring for holding the ring lamp. Okay, and I'm going to connect now the 2 times. Look at this. Of course, I don't need to connect this because it already has an built-in adapter. Yeah, to hold the screws. The reason is because it's so close to the specimen that essentially you wouldn't have enough space otherwise. Okay, so you see that there is already a ring here where the screws of the LED light are able to connect to. So let's have a look here again, stereoscope. So let's connect now the 2 times. And now the distance of the microscope might be a little bit too high, however, okay? That's of course exactly the problem. I have to basically go so close that it might not be possible anymore. Okay, so and for this reason I then have to hold the specimen or whatever I will look at. I have to hold it up. Okay, yeah, so here we go. So now what? I need to kind of hold, maybe I need some kind of a thing to place it on. Okay, let's put it this further away again. Okay, so and I'm going to now put this on here, a little cardboard box, so that I'm able to get the distance higher. And I don't know, let's look at something. I don't know, why not look at the lichens from before. And I'm going to zoom out first, and now this is where it's going to become interesting, because I have to find the focus. I'm already, you see, I'm not even able to go all further down, because I'm already all the way on the bottom position, and I'm not able to focus more, so I need to put it yet higher. Okay, so you know what, I'm gonna add this electrical tape beneath here. Highly improvised, as you can see. Highly improvised. But I just want to show it to you. And let's, so this here is now the lowest magnification. maybe let's center it a little bit here. Okay, with it 2 times, and now let's start to zoom in, and I'm going to keep on focusing so that I don't lose the focus. And you're going to of course see that the contrast and the depth of field really go down, but I just want to demonstrate this to you. You're gonna just try to catch the corner here. Yeah. So that's not a maximum. Okay, as I focus back and forth. Yeah. And very difficult. I'm barely turning it, and I think you get the idea now why. Yeah, if the specimen isn't completely flat, this is going to be very difficult. It's not really useful. Yeah? Yeah. So this is essentially, that's why I never use the 2 times Barlow, because I do not need that high magnification, and the image quality is of course not there as well. Yeah. Yeah. So I'm going to take it off again, and then I see that there are essentially a whole bunch of questions again. So you know what? I'm just going to replace it again, because I do want this to be a little bit more useful. 0.7 times. I'm going to connect it back. No force used. Okay, goes back in here. Yeah, this is actually a demonstration also that sometimes magnification isn't everything. Yeah, I receive emails quite often, especially from beginners who want to know, what magnification do I need? And I say, well, actually, most objects that you want to observe can be observed best at fairly low magnifications. Magnifications are really not that important. Yeah, other aspects like contrast and so on are probably more important. So yeah, here's the leaf again, which we did not look at yet. So let me quickly have a, okay, here we go. Yeah. So let me quickly go through a couple of the comments again. I forgot where I left off. Yeah, the polarized light. Yeah, the make and model. Yeah, it's a Euromex. Nexius Zoom. Silverfish. By the way, last week I found several lacrimaria. Yes, I also talked about those. How often do you find yourself using the stereo microscope compared to the compound microscope? Good question. I mean, I would probably say, well, I'm using my compound microscope more often. Okay, but that is not really a good. That doesn't mean that the compound microscope is always better or anything like it. It simply means that for the things that I observe, I use the compound microscope a little bit more often. The stereo microscope I do use. You'll be surprised a little bit if I've got I don't know, it's got a little piece of a splinter maybe in my skin. Okay, and I would like to observe that, and how do you get it out with a needle? Then I'm using my stereo microscope for that. Yeah. So there are a couple of practical uses in the stereo microscope that the compound microscope is not able to offer. So it is not necessarily a question of how often or on the proportion. There are certain things where you just need to have a compound microscope or a stereo microscope. Yeah? That's simply the thing. I would say that compound microscopes are, if you want to start off with microscopy, maybe compound microscopes are in that sense a little bit more advantageous, because they're more difficult to use. So there are more things that you can learn with them, or have to learn with them, because they're more difficult to use. Yeah. If you want to do something with children, I highly recommend stereo microscopes. Yeah. Yeah, and one thing I also have to say, something that I'm not able to show you here, is actually the stereoscopic impression that you get. So the images that you see here really are nothing compared to the images that you actually see when you actually look through the microscope yourself, because then the objects really jump into your face. Yeah. Yeah. I have the opportunity to add to my collection and buy a used stereo microscope one which is by halogen and one by LED lighting. Both otherwise have the same specs. Which ones would you recommend? Generally, halogen, you see, generally, let's put it this way, LEDs are the more modern technology, okay, and they do have certain advantages, and the advantages is that with LEDs, it gives you a more cool light, okay. So this basically means that when you especially when you are looking at heat sensitive objects, then maybe LEDs are better. However, just a second, I have it here. I do want to show you, however, something. I wanted to make a video of this, but I'm not quite finished yet. But it is possible. It is possible to use to convert quite easily halogen microscopes to LED, and I'm going to show you what I found here. It actually was by the advice of one of my viewers, sent me and that is the following. Look, this is really interesting. Unfortunately, it didn't work for mine. Yeah, but look, these are LEDs, okay, which fit into a halogen socket. So these are basically LED replacements for halogen bulbs. And however, the thing is I didn't quite get the correct voltage, and so it didn't quite work. But that is actually something that you might also want to try out, okay? But with a huge disclaimer, for my microscope, even though I was able to make it fit, it somehow didn't work for one of the microscopes, because the transformer, I think, sensed it tried to adjust it and it wasn't bright enough. Okay, I'm just saying with the other microscope it worked quite well. Yeah, so I'm just saying that these are now also possibilities, but you have to get the correct specifications. Yeah? But it is possible, to convert halogen possibly also to LEDs. Yeah? The reason why LEDs are a little bit easier for photography is because in halogen, when you change the brightness, you're also changing the color temperature. So if you turn down halogen, then the light becomes more red, and this might not be an advantage. So you have to do the white balance again. Yeah. Can you see microorganisms directly? Look, I want to show you something, not on the moss sample, but I do want to show you some algae here, because I was barely able to see, indeed, some microorganisms here. Okay, and not very well. Okay, so I'm going to turn off this from the top, from the bottom, and I'm going to turn on from the bottom. Yeah. So what you see here on the top, the move, that's not a per definition not a microorganism. The thing that's moving all the way on the top, it's a water crustacean. I think it's an ostracod or something. Okay, so per definition, strictly speaking, not a microorganism. You can look at it in any case. Okay. But if you want to look at individual cells, stereo microscopes are probably not the best, you see, to use. Okay, let's just stay here with the algae. I have to focus. I have to find the focus. There is a little lag here as well. Okay, so let's go up a little bit with the brightness as well here. Yeah. So there might not be a lot of single cell organisms in here, and in any case, yeah. But if we move on, go yet higher, yeah, that's the highest that we're able to get. Okay, then this is basically what we're able to see, right? And you see that that the contrast itself is also not very high, so there's lots of stray light as well. Yeah. But I have seen in this sample a couple of rotifers and so on. But for single-celled, would you be able to see single-celled, let's say paramecia or so? Yeah, sure. But you're not able to look very well into the cell. Yeah? So the question is, are you able to see them? Yes, but are you going to be happy with the quality of that's a different thing. Yeah? So you see, stereo microscopes are really not designed to look at some of the specimens. They're really good at for looking at opaque specimens. And many microorganisms, ciliates, whatever, are transparent, and therefore they are difficult to see. Okay. Yeah. So it's really a question of what are your quality considerations here. Okay. So to my knowledge, it's not hard to reach 10x with a stereo, but more than 10 times is difficult, expensive. Yeah. Lens, yes. So I'm again reading through the comments here. I think stereo is much better for beginning the hobby. I find compound very complex relatively in terms of sample preparation and so on. Yes, yes, and yes and no concerning sample preparation. I'm saying no because unless you buy, of course, finished specimen slides. Okay, so if you do not want to prepare samples or so, then of course you have to fall back on prepared specimen slides. Yeah, yeah. It's used a lot in electronics. Yes, there are, of course, stereo microscopes who are on this boom. Yeah, so with basically so that you don't have a stand like this, but they're on a boom, so they're connected to the table, so that you actually have your tabletop, your desktop for soldering and for doing microelectronics. Yeah? Okay. Yes. What is the difference between LED and halogen? Well, let's put it this way. Halogen is the LED, well, from a technological perspective, halogen has a glowing filament in there and generates a lot of heat. Okay, but it can also maybe be very bright. However, in recent years, the LED technology has really caught up, and therefore, for concerning brightness, the LEDs are equally bright as the halogen. Yeah, so that is the thing. So it's a question of technology, I would say, and yeah, mostly of technology. Yeah? I use my stereo microscope to ensure my biological microscope optics are clean, especially the one. Yes. So what you can do is you can take, for example, optics, microscope optics, and put them under the stereo microscope, and also check the quality checks as well. Okay. So you know what? I'm just going to put a few more things. Yeah, under the microscope. What time is it? Ooh, it's already 1 hour. I still have a few things here. Yeah, let's do the following. Here I put a moss sample. Just before I started the live stream, I put this moss sample here. Okay. And there is indeed a bit of movement, as you can see. Very difficult to see, but so there might be. I have actually seen a few rotifers here in this sample. Yeah? And the image quality is also not so good because some of those microbes or microanimals are kind of hidden behind the moss leaves, but the movement that you see here, I mean, that is essentially, yeah, it could be microorganisms. Let me just quickly check if my my compound microscope is working. Seems to whether the connection is working. Yes. Yeah, the circle in the stage I'm gonna remove. So here we go. So I'm going to now use this here, and I'm going to put it now into my compound microscope so that you see the difference. Okay. Okay, so I think you get the idea that there is a huge quality difference. I'm going to remove the DIC filter. Yeah. Let's go down with the magnification so we have almost the same magnification. I need to swing out the condenser. Where's the condenser? Here it is. Okay. Yeah. So that is the moss now, yeah, under my compound microscope. So you see that, yeah, for viewing, of course, individual cells, microorganisms, and so on, for viewing transparent objects, let's put it this way, the compound microscope is of course much better. Look, there is a tiny rotifer, yeah, almost in the corner there, yeah, top left, some movement top left, yeah. Seems to be a rotifer. We can move in, zoom in a little bit more. So now it's in the center. There seems to be some kind of a a disturbance. There is some, I don't know why. Yeah. Yeah. So, yeah. And just very quickly, we remember where it is. I don't know if we're then able to also find it again here under the stereo microscope for comparison. This is difficult. Yeah, I don't know. Okay. Yeah, I have to find it again. Don't know where it could be, here somewhere. Or here, I don't know. Yeah, you get the idea. So, stage movement in the compound microscope is opposite. Yeah, the thing is the following, that in a stereo microscope, what you have here on the top in here, you've got prisms which rotate the image right side up. Okay, and the reason why this is necessary is because if you want to manipulate the specimen and move it around, then yeah, it's like a normal extension of your eyesight. Yeah, so everything is right side up and you see three-dimensionally. In a compound microscope, it doesn't really matter how the slide is positioned, because you cannot see the original anyway because it's so small. Yeah? So whether it's upside down or right side up really doesn't make a huge difference, because you don't know it anyway because it's so small. Yeah? So this is the reason, but for stereo microscopes, it's that's important. Yeah. Yeah. So yeah. So next thing, I talked about how I want the algae I showed you. Let's have a look at this here. This is some fur, I think, either from a dog or from a cat. Now I remember. Okay, so some cat fur. Yeah. I made some time ago. I did make I think I showed this. Might have showed it to you already, right? Some bubbles. I made a permanent mount. Yeah, yeah. And when you zoom in, yeah, then this is how it looks like with light coming from the bottom, and this is how it looks like with a light coming from the top. And because there is this clear glass plate here, let's see how this looks like when I put it on a mirror. Okay, almost has a little bit, looks a little bit like dark field, and again gives it quite a different characteristic. But actually, yeah, there is too much stray light, so the contrast is not really quite as good as I expected it to be. Let's use the other lamp. Yeah. You see, because of the glass. So what I would suggest maybe sometimes, and I don't know if this actually works, is maybe by lifting it up, and you're not able to see this now. Yeah. By lifting it up. This is also not going to, not gonna work. Okay. So turn it up like this here. I'm experimenting and trying to get the background dark, but as you can see, I'm having some focusing issues here. Here we go. Yeah. Okay, well. So I lifted it away a little bit from the mirror. Yeah. In any case, there are plenty of things that we can experiment around with. Yeah, I'm slowly thinking about finishing, but I do want to show you some sand samples as well, because these are actually quite nice. Yeah, that's one of those slides that I made some time ago, and essentially, I need to explain this if you have not seen those before. Those slides here, yeah, there are two of them here. Yeah. And if you look at it in here, the sand is actually able to move in here. These slides are concave slides, and what I've done is I used some nail polish, some blue nail polish, and that's how I glued the cover glass on top. That's why it's blue here. And as the nail polish started to dry, it started to pull in some air bubbles. But the nice thing is that this system allows you to look at the sand in a very native way, without any mounting medium, surrounded by air. Yeah, and let's have a look here. So that's actually also a nice way to make certain sand collections. So let's go up a little bit, maybe more light. Yep. Okay. Yeah, so that's basically, yeah, that's basically some sand under the microscope using a sand slide, which allows the sand grains to move. So a second way of how you can make sand slides, and I made this a few weeks ago and now the glue has dried, is by simply gluing the sand on a slide using simply some glue. Again, I'm going to go zoom out. Yeah. So I spread some glue over the slide and sprinkled the sand over it, allowed it to dry. Has the disadvantage that essentially the sand grains are surrounded a little bit by glue, which you're able to see here. So it's not quite as natural appearing as as the other slide. Yeah, but still, yeah. So some people say they can't hear me very well. Okay, maybe the microphone has to be moved a little further to my mouth. Okay. So I just wanted to say that this is basically, there are different ways of preparing sand slides for stereo microscopy. And if you were to look at these slides now under the compound microscope, then yeah, it would look a little bit different again. So I've lost it. Here we go. Yeah. Is there a way to polarize light for stereo? Yes, this is the, okay, this is something I also wanted to show you. Yes, there is. And I would like to show and demonstrate this to you, okay, as a last thing. So a little bit of simple polarization microscopy. What you need is you need two filters. Let me show this to you on the, how do you say this? On the desk view. What you need is you need two filters. Now I've got some linear polarizing filters. They're in glass. However, you can also get plastic filters. For example, I got those here from 3D glasses. I do not recommend those because they're circular polarizing, but you can actually buy them in small plastic sheets. And what you do is you place one of those filters on the stage, okay? You need to have now light coming from the bottom. That's important. Yeah. So this is now, again, you have a problem seeing it a little bit. Okay. So I placed one of the filters here on the stage. Let me turn down the light a little bit. Yeah. And then you place a specimen slide, and I put a little bit of vitamin C on here and crystallized some vitamin C. Honestly, it's not the nicest slide, okay? But it's simply a little bit of a proof of concept. Yeah. Why vitamin C? Because normally it makes beautiful crystals, but in this case, not really so interesting because maybe I didn't get the concentration right. Okay. So you can now see the crystals, right? However, you need to now put the other one over it. Okay, maybe you can check again here on the top. I'm putting it over, and I have to rotate the filter until the background becomes completely dark. Okay, like this. And because I do not want to put it directly on the crystals, I'm going to use this spacer which I made. Okay, this plastic spacer here, which I also used for my mobile phone camera. And it works also quite nicely as a spacer. So I'm just going to put it on here, and I'm going to rest this over here. I'm rotating it until the background becomes dark. And then, of course, I'm going to refocus. Yeah. Now, it doesn't look quite as nice because the crystals look a little bit too bright, and that is the reason, again, because of the camera exposure. You know what, I'm going to try this. I hope that this is going to work. Let me quickly adjust the exposure time a little bit by making it a little darker. I don't know if this works or not. Deactivate, I have to activate it again. I hope that this works. Not so much. Okay. Yeah. But I think that's simply a problem with the exposure time now. Yeah. So this is basically now polarized light. Yeah. stereo microscopy. Yeah, it works, and I would say it's probably easier than probably with a compound microscope, because there's simply much more space for those filters. Theoretically, what I could try to do is why not? Let's try the same thing, the polarization with the sand grains. Why not? I've not tried this yet, I'll be honest with you, so I don't know what to expect. I'm just going to put it directly over it. Okay, a little bit lower magnification maybe. This, yeah, it's, I think some of the sand grains simply depolarize the light. Looks a little bit like dark field. Okay, yeah, not so. Yeah. Let's try the different. This is the second sand sample. Ah, a little bit of color. Very nice. Turn up. Okay. And let's try to zoom in a little bit, or zoom out. Yeah, there seems to be, depending, maybe some of the sand grains are a little bit crystal. Yes, it's very dark because I forgot. Yeah, I didn't forget. I made it darker. So just a second, I'm gonna go up a little bit with the exposure time again. So maybe it's gonna be a little bit brighter. Deactivate, and I have to activate again. Yep. Okay. Yes, that's I think a little bit better. So you see that some of the sand grains indeed show those rainbow colors. So there seems to be some, seem to be optically active. Yeah. So I'm going. Still looks pretty nice. Yeah. Is it only the polarization filter? I'm actually using two polarization filters, one on the bottom, then the specimen, and then one on the top. I can actually show you the orientation by going back to the desk view. Okay, so you need two identical polarization filters. Okay, the one directly over the lamp, then you put the specimen over it, maybe a spacer or not. Yeah, and then the other one, and this one you have to turn until the background becomes dark. So in the crossed position, the light is blocked unless the specimen depolarizes it. Okay, and if you do not, and if you don't want to get those, then get yourself linear polarization filters. You can buy them on Amazon. They're not too expensive, and then you basically can cut them into shape. I would say do not get don't get those for the 3D paper eyeglasses, because these are circular polarizing filters, and then you also have to get the correct orientation. Back front is important, and this simply makes it more complicated. Just get regular, simple, linear polarization filters. There are some very cheap USB microscopes that seem to reach. Okay, cheap USB microscopes. What are your thoughts on them? Look at what I got here. I wanted to demonstrate this today, honestly, because I have 1, 2, 3, 4, 5. I've got 6 USB cameras connected to the computer right now. For this reason, my computer was not powerful enough for another one, so it always kept on disconnecting. I bought a couple of years ago, I think I directly bought it from China. I think I paid €7 for this microscope here. It was ridiculously cheap, so don't spend too much on those. And as a matter of fact, these microscopes will give you pretty much the same view as you get on a stereo microscope without the 3D effect. So you can connect it directly to a power supply, USB. Not power supply, but USB. I'm gonna do this in any case now, simply to show to you how this actually works. Okay. However, I unfortunately was not able to actually use it. And then if you turn this here, then it should actually work. And see, it doesn't even work because my USB power is, for whatever reason, not working. Interesting. Okay, it used to work before, but essentially there are LEDs in here, and there is a lens in here, and by turning this, you can change the focus. It says, you know what it says here? 1,000 times forget it, never. Okay, I don't know what they've been thinking. But it allows you to focus, and you see it kind of moves in and out. And you just basically put it over whatever you want to look at, your skin. yeah, sand, and it works surprisingly well. Okay, so considering the fact that they're so cheap, and if you have some thoughts of should you buy yourself a stereo microscope or not because of the cost or whatever, I mean, you just spend a couple. Don't spend too much on these things. I spend around seven euros on this. It's $8 or something, $10. And they're really cheap. I'm not saying it's the best quality. It kind of does its job, okay, and it's good, quite good for actually, yeah, playing around. Yeah, just saying. Also, if you have kids maybe and you want to explore the world, yeah, then you can use those. Yeah, yeah, I have the ones mounted on the microscope. I mean, filter and analyzer. What you need is, filter. We call this the two things here. One, even though they're the same. The first one is referred to as the polarizer specimen, and the second one is referred to as the analyzer, but actually they're the same. Yeah. It was once again very educational and interesting. Thank you. Yes, and I think I'm also slowly starting to feel a little bit tired. Yeah, I started to sidetrack a little bit today as well, but I'm just going to leave it at that. But I'm going to, yeah, simply show you maybe some of the other samples later on, but actually most of the things I already showed you anyway. Yeah, and hope it was indeed informational for you. Concerning next week, important announcement. I will try something different. I will try to pre-record something, and then it's called a YouTube premiere. So this basically means I will play a pre-recorded video. And you should be able to also interact in the chat, and the reason is because for business reasons I'm not around. Okay, for school related reasons I have an appointment, and I still want to use this opportunity to still have a YouTube video next week, but one which allows you to interact with the community by posting chats like you're doing right now, but I will not be able to answer them because it will be pre-recorded. I've never tried this before, okay? So I'm gonna see if this actually works. But for today, I'm going to, unless there are some last second questions, I mounted polarization filter on the surface of an eyepiece, and it gives a bit of a different effect with colorful background and a bit of polarization effect. The reason when you mount a polarization filter over an eyepiece is it sometimes it gives those rainbow colors, is because the optics of the eyepiece are not completely strain free. So there are some tensions inside the glasses, and they cause the light to become a little bit depolarized, and sometimes this is the reason why there is some color effect as well. Okay, all the best. So this means no live stream next week. Well, I do want yes and no. I will not be personally interacting, but I want to actually play a video where essentially, which appears to be like a live stream, but which basically where you can use the chat box on the side. Okay, so I've not tried this yet before, but we're gonna see if this works out or not. Okay, so I wish you all the best. Happy microbe hunting. See you around next time. Bye bye.